Hello, everyone, and welcome to this webinar, which is hosted by Tissue Engineering and Human Gene Therapy, two of the more than 100 journals in the Marianne Liebert publishing portfolio. Today's subject is the impact of a cage change event on in-cage activity and microenvironmental conditions in IVC, individually ventilated cage housed mice. Today's webinar is sponsored by Techniplast. Thank you very much for joining us today. I am delighted to serve as your host and moderator for today's webinar. I'm Kevin Davis, the founding executive editor of the CRISPR Journal, as well as a new marquee peer-reviewed journal from Marianne Liebert, Gen Biotechnology, publishing original research papers in all aspects of biotechnology. I'm also the author of Editing Humanity, the CRISPR Revolution and the New Era of Genome Editing, which is available in audiobook and paperback. Today, we will be hearing from a trio of experts, all of whom are co-authors of an important study that was published last year in the open access journal PLOS ONE, and that will be a key subject as we go forward today. The subject of that paper was a multi-center study on spontaneous in-cage activity and microenvironmental conditions of IVC-housed C57 Black 6J mice during consecutive cycles of biweekly cage change. And here to discuss that and other aspects of that study and this area of research are three of the authors of that study. We'll be hearing from Dr. Jan Bas Prins, who is the director of the Biological Research Facility at the Francis Crick Institute in London and professor of laboratory animal science at Leiden University in the Netherlands. He's also a member of the Dutch National Committee for the Protection of Animals that is used for scientific purposes. He will be joined by Dr. Hervé Lerat, professor at the University of Grenoble and manager of the core animal facility there. In 2021, Hervé was named director of the newly created Unité de Services HTAG at INSERM. And last but not least, Dr. Brun Ulfaker is emeritus professor in anatomy in the Department of Laboratory Medicine at the Karolinska Institute in Sweden. We are delighted to have them all with us today. For this webinar to be a success, we really want to hear from you, the audience, so we would love to get your questions. Please enter them at any time using the Ask a Question box on your screen, and we will put as many of them as we can to the expert panel at the conclusion of their advanced presentations. So we look forward to your input. And so we are ready to begin today's webinar. The first of three presentations will be from Dr. Jan Bas Prins from the Francis Crick Institute in London. Dr. Prins, great to have you with us. Over to you.
Dear all, my name is Jan Bas Prins. I'm one of the investigators who participated in this multi-center trial on the spontaneous in-cage activity of C57 Black 6 mice. And I would like to give you an introduction to our studies that those were published in PLOS ONE, and it was a multi-center study on the spontaneous in-cage activity and microenvironmental conditions of IVC-housed C57 Black 6J mice during consecutive cycles of biweekly cage change. And as you can see, there were multiple centers involved, like the Karolinska Institute, with Brun Ulfaker as the leader of that center, the Institute of Molecular Genetics of the Czech Academy of Sciences, which was led by Jan Honigschlager, the University of Grenoble with Lerat, and then there was the Leiden University Medical Center in the Netherlands, which I was the leader of that section of the multi-center trial. So the main objectives of our studies were to determine activity level dynamics of group-housed mice who were undergoing a standard IVC changing regimen of once every 2 weeks. And the second was to determine the ammonia concentrations dynamics in those IVC cages during those 2 weeks in the cages under flow conditions, which is quite different from many other readings that you will find in the literature. And then the third main objective was actually to determine the areal spacing dynamics of activity, so whether the animals were spending less or more time in particular areas, and whether there was any dynamics during the 2 weeks between cage changes. So we used for this study, we used the DVC technology as developed by Techniplast, and each of the centers had Techniplast DVC racks. And what the DVC actually does is it has, under each cage there is a sensor plate, and it's an electromagnetic plate with 12 different sensors in it, and these sensors actually operate the same way as, for example, the screen of your device, your telephone, your smartphone, for example. So when you push your finger on the screen, it will change the electromagnetic field in that area and actually gives a signal. So here you see how that works. The plate itself, without any cage or anything in position, you see as the highest level of that particular sensor is the capacity of that sensor. So that's the no cage level. As soon as you put a cage in, then you see the capacity drops a bit, and then actually a signal is given. Okay, there is a cage in place, and this is a cage with dry bedding, for example. If then a mouse crosses that particular sensor, then you will see there's another depression. And once the mouse leaves that particular sensor, again, the capacity goes up. So that is how you measure and how you follow whether there is a mouse present or absent. And in that way, you can actually find the activity, you can measure the activity and also the direction of the move. And if a cage gets wetter because of the increasing urination and the accumulation of turds and everything, then you see that the cage gets wetter, and you see the capacity dropping, and that actually means that we can detect through the DVC system as well whether a latrine is forming. And we have actually within the study done both a visual inspection and identify where the latrine was being made, and the other is going by the DVC system and see where the latrine, according to the system, was developing. Now, in order to determine where most activity was spent, we divided the cage into a front and a rear. So the rear were the sensors 1 to 6, and then we have the sensors 7 to 12 defining the front area, and that's how we assessed whether activity was more in the front or in the rear of the cage. This is an overall view of some of the material methods and also how we worked our ways through the cages and how we positioned the cages in the rack. So all the studies were done by C57 with C57 Black 6J animals, males and females. And we started the experiment with animals in the age range of 6 to 8 weeks. We put 4 animals per cage, so a maximum of 4 animals per cage. And the bedding was an aspen bedding, about 100 grams per cage. As cage enrichments, we used Better Nest and a cardboard house. And the diet was SDS-R3 irradiated diet. There was a 12 light cycle, and the temperature was within the range of 20 to 24 degrees, and the relative humidity 45 to 65%. Every other week we did a cage change, and also every other week we weighed the animals. And before we started the study, there was 2 weeks of acclimatization of the animals. Now, when you look at the right-hand panel, you see there an image of the rack, which has 6 columns of 10 cages each. So when you look at the left-hand and the right-hand side, those were the columns that we actually used for the study, because we did ammonia lab measurements under flow conditions. So we couldn't use the inside cages. We could only use the cages at the left and the right side column. Every cage, and the blue cages are the male cages, and the orangey type cages are the female cages. And every other week with the cage change, we also changed the position of the cages. So we moved the cages down, and then the bottom cages weren't all the way up. And this was to eliminate the effect of light as a confounding factor in the study. So the ammonia measurements that we did were as follows. So as I said, the ammonia measurements were done under flow conditions, which is quite different from many other studies. And we did it by leaving the cage, obviously, in the system, connected to the ventilation. And as you can see on the in the A panel, you can see that on the side of the cage, just above the bedding, there are holes that, in this case, are stopped with a plug. So there's one in the front, one in the middle, and then there's one in the rear that you can't see on this image. We would unscrew one of those plugs and then put the probe in, and the probe was a rod that was connected to an ammonia meter, and then what you see is that the rod had holes that cover the entire width of the cage. And we would measure that during a particular period of time to allow actually the system to equilibrate to whatever the measurement was, and then record it. And we did that through all the three positions of every single cage. So here's the protocol for all 4 sites. That is, at week -2, animals were put in the cages in a randomly. And then on week 0, week 0, the whole experiment started with a cage change. And then, as you can see, every other week there was a full cage change, and the ammonia measurements in between those 2 cage changes were done at day 2, 4, 6, 8, 11, and 13, and 14, obviously, the day of the cage change again. So there was quite a number of measurements. The numbers were close together at the final few days in the cage change cycle. Now, there were some additional experiments performed at the Karolinska Institute by Brun and his colleagues. There was a measurement of the in-cage bacterial growth in between cage changes, because we were quite interested in whether the bacterial growth in between the cage changes was substantial, and actually also reflecting the ammonia production that we measured. So in order to do that, we swapped the cages, all 4 walls and corners. And at the end of 5 bi-weekly cage changes cycles and the end of 2 cycles of weekly cage change. So we compared actually what happened after 2 bi-weekly cage change and what happened after a weekly cage change, and compared those. The other thing that was done at the KI was a post-hoc histopathology examination, and that was actually to assess whether there was any pathology visible in the airways of animals because of the exposure to ammonia. And in order to do so, we also needed a control group. And the control group, we choose a germ-free colony of C57 Black 6J animals. And the third additional experiment that was done at the KI was actually the impact of bedding material. As I said earlier, all the 4 centers used aspen bedding. At the KI, I did an additional experiment comparing aspen with corn cobs. You can see here how that was done. The aspen chips were in 10 cages with 4 males, and the corn cob in 10 cages with 4 males. And we did the males because they typically urinate more than the females. And that was done in 3 consecutive cycles of bi-weekly cage changes. And then it was also done by looking at reduction of the density of the animals by reducing the numbers to 2 animals per cage, and again 2 consecutive cycles of bi-weekly cage change. So here there is a comparison between the aspen and corn cob with different densities of animals per cage. Now, the data processing and data computation is that the DVC data were aggregated in bins of 1 minute. So we got activity data throughout those 10 weeks for all those animals aggregated in bins of 1 minute. And the measurements were obviously done during the daytime activity lights on and the nighttime activity lights off. And we also looked at the percentage of daily activity performed during the lights on period compared to the overall activity. And the activity distribution, as said, in the front and the rear of the cage, and how that changed. And that was expressed as frontality. So that's the percentage of activity performed in front of the cage, in the front half of the cage. And then we looked at the latrine position as well, relative to that activity, just by visual inspection of the cage, and also at reading the capacitance of the DVC system. The statistical analysis was a repeated measures analysis to test for the differences across sites or sexes, and also the cage change cycles and days. And since we could not, there was not always a ascertainment of normality, a non-parametric test was chosen, the Mann-Whitney test and spearmint rank correlation tests. And now I hand over to my colleague Hervé Lerat, who's continuing this presentation on the study by presenting more. Thank you very much.
My name is Hervé Lerat. I worked at the University of Grenoble in France, and I will present the data we gathered on animal activities during using the DVC racks and the experimental design that Jan Vaz just described. The DVC technology allowed us to record the animal locomotion activity 24/7. On these graphs are reported the normalized activity in the 10 cages during the entire study and on the 4 sites involved in the different colors here. The x-axis represents an entire day, showing the lights on period and the lights off period. Lights on here on the left, in white, and lights off here in gray on the left. The C57 black sea mice that we use, they show a characteristic and reproducible circadian rhythm, 2 lights on and lights off. This is expected for nocturnal rodents, and moreover, you can see easily here with the different colors showing the different size, that this was very reproducible across sites with some moderate differences. Here we averaged the daytime and nighttime activities across the different 5 cage change cycles for males in blue and females in orange. Again, lights on is in white here, and lights off are represented by gray stripes. The 4 different sites where the study was performed are shown here. During daytime, we observed no difference here, or here, for example, between sex, but differences between facilities and also some differences across cage change cycles, which is not shown on this graph. During the nighttime, lights off, we observe highly significant differences between sex here, male and female, you can see, and some differences also between sites. Next, we studied the impact of the cage change activity on the animal locomotion activity. These heat maps that are shown here, they display locomotion index. And the locomotion index is an average of all plate sensors of the DVC. And this during the entire day. This is shown at the bottom here from 0 to 24 hours. And across one entire change cycle, 14 days, y-axis, here, 1 cage change cycle, 14 days. The darker the color, the blue color, the less activity. Again, light cycles are shown on the top, lights off, lights on, lights off again. Again, the circadian rhythm is easy to see. A lot of activity during the night, less activity during the day. And here I highlighted in red ellipse the cage change. Here you see cage was changed here that day, was changed again that day, and same thing here for the females. Day of change, day of change. What you can observe just after the cage change, the days after, you can see that the activity is perturbed for the mice. It's easily seen here for females, for example. What we've done, we quantified this cage change-induced perturbation and plotted them on these graphs. The average activity on the y-axis and the cycle day on the x-axis, again 0 to 14 days, corresponding to 1 cycle of cage change. So the average activity was calculated using the data of 5 different cage change cycles. Again, the male in blue and the female in orange. Lights on top of the graph here, and lights off at the bottom. We can observe that cage change, they induced a marked increase in activity, especially during daytime. The difference could be almost 50% when the animal are supposed to rest. This burst was then followed by a gradual decrease of activity, as you can see here, also for the females, till the next cage change. Then we analyze the use of the cage floor, especially the latrine area preferences. This bar graph shows percentage of latrine in front, in white, or rear, right here, or rear in gray. This is according to the sex, the males in blue, females in orange, and according to the 4 different sites where the study was performed. We've observed that the male mice, they showed no real significant preferences for creating the latrine in the front. It's like 60% in the front and 40% in the rear, so averaging that 50%, even though the food, the water, and the shaded area were present towards the rear of the cage. In contrast, you can see that the females mice, they prefer to have the latrine in the front of the cage with an average of about 74%. Now, what we've checked was the percentage of latrine at the same spot after each change. And here, what we are showing, this percentage is when the latrine is unchanged. So at the different sites and according to the male and the females. What we saw here is across cage change cycles in a given cage. The males in blue here are prone to change latrine localization compared to the female here, where they used to keep more often the latrine at the same spot after the cage change. Okay, knowing the latrine and the position of food and water in the cage, we then analyze where the animals show preferred activity on the cage floor. For this, we analyze the locomotion activity data from the 6 front sensors here. We pull the 6 front sensors activity, or for the 6 rear sensors, the activity was pulled for these 6 antennas, and we calculated the frontality percentage. The frontality percentage, actually, it's the percentage of time that the mice spend for the locomotion activity on the front area of the cage, here, presented in blue here. Okay, so here you see these different graphs. You have the frontality on the y-axis, and again, the cycle day between 0 and 14. So 1 entire change cycle represented here, an average between the 5 different cage change cycle that we've performed for this study. Okay, so what did we see? We saw that frontality progressed through the change cycle. Okay, it's not stable, it progressed. But overall, what we see is the male mice, they allocate more of the activity in the latrine-free part of the cage. You can see here, in blue here, we analyzed the activity when the latrine was in the front of the cage, okay? And you can see that the frontality is decreasing, is about 50%, meaning that they spend more time on the rear of the cage and not in the front. And also, when the latrine is on the rear, in green, you can see that the activity, the frontality is going up and is high, meaning that they spend a lot of time in the front of the cage at the opposite side of the latrine. Same analysis for the females, but different results. Indeed, the female mice, as you can see, they use the entire cage floor the first week, okay? This was pretty much true across all sites where we studied that. And during the second week, then we see a decrease in activity at the site of the latrine. Okay, so same behavior as the males. Okay, then we tested if this behavior could be impacted by the type of bedding or the number of mice in the cage. Both parameters having important impact on the latrine area. This was tested on males. These are the results we are showing here. They've been done on males. We compared here on these two graphs, for example, aspen bedding that was used throughout the whole study with corn cob. And we formed four mice, male mice in the cage here. As you can see, the avoidance of the latrine area was not impacted by the type of bedding. Again, you can see when the latrine is in the rear, the mice, they spend more time, frontality is going up and is high here in green, and the opposite when the latrine is in the front. You can see that the males, whatever the bedding, are spending less time for their locomotion activity in the front, more time in the rear. We also checked the number of mice, the impact of the number of mice, like I said. So we compared 4 mice, 2 mice, and actually in the two cases with the two different bedding. As you can see, reducing housing density to 2 mice completely abolish this behavior of avoiding the latrine area. So I thank you for listening, and now Brun will present the next part of the study.
So the next step is to try to see to what extent the position of the latrine correlates with the in-cage levels of ammonia. And to localize the latrine in each cage and in each cage cycle, we use the DVC sensor board. And the readings from the different rows of electrodes you can see in the panels here, and the larger the drop in the sensor that we could record, the more likely it is that we have the latrine there. So this shows an example of the E3 cage. So we use this to localize the latrine for each cage, each caging cycle. And then we plotted the values of the ammonia concentration, which we obtained through the 3 different probe sites across the cage floor, the front, the middle, and the rear. And we plotted that against the presence of the latrine, if it was in between, and the area opposite to the latrine. Females are in red, males in blue. And as you can see, the ammonia levels across the cage cycles are higher in the latrine area than in the area opposite to the latrine. And this replicated across all 3 sites, so we saw the same pattern. In addition to that, you can see that the blue tend to be higher than the red, so males usually display higher values for the ammonia concentration inside the cage. Now we can summarize this that ammonia concentration is significantly higher in the latrine cage floor area than in the opposite area. Ammonia concentration is significantly higher in cages housing males than cages with females. Next step was to, in greater detail, analyze the buildup of ammonia concentration across the cage change cycle day by day. And we used then the same setup here, where we have localized the latrine to the cage front, to the cage rear. We have males in blue and females in red. And this shows an example for one other side. We can see on the x-axis here the days following the cage change until the next cage change. And here we have the levels of ammonia. We can see that the levels of ammonia rises from 0 to about 50 at most across the cage change cycle when the latrine is in the front. The area opposite to the latrine, over here shows also an increase in ammonia levels, but not to the same extent at all, as we can see in the latrine area. This pattern replicated across all 3 sites, so we saw the same pattern. Next, we analyzed when cages and caging cycles where the latrine was in the rear instead of the front. And we saw the same principal pattern across all 3 sites with a buildup of ammonia concentration across the caging cycle, females in red, males in blue, and that the levels of ammonia also increased in the area opposite to the latrine, but to a lesser extent in the area opposite to the latrine. You can also note that there is a difference if the latrine is in the rear of the cage and the front of the cage. So we reach higher values for ammonia concentration towards the end of the caging cycle when the latrine is in the rear compared to when the latrine is in the front. So we can conclude that across the caging cycle, the ammonia concentration increases from 0 up to about 25 ppm in the latrine-free area and up to between 50 and 100 ppm in the latrine area at the end of the cycle. Measurement of ammonia concentration across the cage floor, as I showed here, revealed 3 times higher values for the latrine area compared with the opposite area. Ammonia levels was higher with the latrine in the rear, and this is possibly due to the airflow dynamics of the IVC shown here. So the blue lines here and arrows indicates where fresh air comes in, and the red is where the air is getting out of the cage. And this is likely the explanation to the difference between latrines in the rear and front. We then moved on to do some more detailed analysis in a separate set of experiments where we asked if housing density, bedding material, and latrine position impacted ammonia concentration inside the cage. And we have the same setup with latrines in the cage front or in the cage rear. We have 2 sets of cages, all with 4 males inside the cage, and one set, blue denoted with blue color, has aspen chips as bedding. Those with green colored circles have corn cob as bedding. And we have the number of days post cage change on the x-axis, and here we have the concentration of ammonia. And we can see that what we saw in the multi-center study replicated well in this set of experiments. So we have an increase in both sets of cages with ammonia concentration. And this increase is higher in the latrine area compared to the area opposite. So what we see here is a buildup of the ammonia concentration across the cage change cycle for both bedding materials, and it's higher in the latrine area compared to the area opposite to the latrine. With the latrine in the rear, we see the same pattern, but again, we replicate that with the latrine in the rear, the ammonia level will reach higher values than when the latrine is in the front. Next, we ask if we now change the density of housing. So we move from 4 males per cage to 2 males per cage. And this is the readings of this experiment. And as you can see with the latrine in the front, there's practically no ammonia across the cage change cycle, regardless of bedding material. With the latrine in the rear, at the end of the cage change cycle, we could record values between 20 and 45 ppm on both bedding materials. It's also important to note that we tried with statistical testing to see if there was any difference with the starting values and the end values, depending on which bedding material we used. But these sets of data do not reveal any statistical significant difference in ammonia levels at the end, in the beginning and at the end of the cage change cycle, regardless of bedding material. So housing density is a significant factor for ammonia concentration buildup during a cage change cycle, while the choice between aspen chips or corn cob as your bedding material appears not to be. Mice do not excrete ammonia. They excrete urea. And urea is broken down into ammonia and carbon dioxide by the enzyme urease. Urease is produced by bacteria colonizing the cage. So we asked, is there a correlation between the degree of bacterial colonization inside the cage and the level of ammonia inside the cage? And we had two sets of cages, one with weekly cage change with females, open circles, and males, and then filled circles with biweekly cage change, females and males. On the x-axis, we have bacterial load in colony-forming units, and on the other axis, ammonia concentration. As you can see, there's a clear relation between the colonization of the cage and the level of ammonia that we can record from the cage. Surprisingly to us, we saw not a statistically significant difference in the colony formation inside cages with biweekly compared to weekly cage change. So if we summarize this, as expected, in-cage bacterial load co-varies with the in-cage ammonia concentration, and bacterial colonization is already saturated after approximately 1 week. We then asked if the level of ammonia that the animals were potentially exposed to caused any harm to the upper airways. So we did histopathology of the upper airways, and as reference to compare with, we used germ-free mice. Germ-free mice are never exposed to ammonia because they live in a bacterial-free environment. So these animals served as a reference to our experimental animals. Guided by the literature, you can read here, we selected 9 variables in 4 different localities to be assessed. And the assessment was made by 2 trained histopathologists on coded slides. And they graded the presence of stigmata on a scale from 0 not present to very abundant, giving a 5. This is the outcome. Again, I will not go to any details with respect to locality and the variables. I will only I will move directly to the fact that only 4 of these 9 variables here in bold could we see a difference between our experimental animals and those animals that was brought up without any exposure to ammonia. We then asked these 4 variables, how the ranking of these 4 variables, the presence of the stigmata in the different animals, how did that correlate with the in-cage measured level of ammonia, bacterial load, and sex? And to do this statistic, we used a mixed model principal component analysis shown here. And I will not go into the details here. I will only help you to get the bottom line out of it. We can see that the variable sex, ammonia concentration, bacterial load, all co-vary strongly and loaded on the F1 axis. One of the histopathological variables, inflammatory cell infiltrate in the olfactory epithelium, loaded only on the F2 axis and showed that absolutely no correlation with these variables. The other 3, which was statistically more prevalent in our experimental animals compared to the germ-free animals, they were localized in between. So they showed kind of weak covariation with ammonia levels, bacterial load, and sex. Altogether, the set of data here analyzed could only explain 45% of the total variance in the data set. So we can conclude that the statistical assessment revealed that there was very little or no covariation between the grade of histopathological aberration and inferred ammonia exposure. Furthermore, the low degree of covariation and the observation that several variables were equally present in germ-free and experimental animals suggests that these variables are caused in full, or at least partially, by other in-cage parameters than ammonia. Now, I'll come to the summing up of our key results from this study. And home cage pattern of activity and rest is strictly coordinated by circadian rhythm. Females, however, are more active than males during nighttime. A cage change induced a marked hyperactivity in both sexes, a behavioral disruption that faded off in the next following days. The hyperactivity is most marked daytime and will thus disrupt the natural sleep-rest pattern of the mice. The effect size of the cage change on rest and activity is so large that it should be considered as an experimental variable and included when designing the experiment. Mice allocate more of the activity to the latching-free part of the cage floor. This behavior progressed through the caging cycle, according to sex, but was not impacted by the type of bedding used. We observed an ammonia gradient in the cages with higher concentrations over the latching area. Ammonia concentrations in the front of the cage were lower than those in the rear of the cage, irrespective of sex, housing density, or bedding type. The absence of marked histopathological alterations in the upper airways and the low degree of variation with ammonia level and bacterial load is not surprising, considering the results of controlled exposure studies. A consensus on an evidence-based exposure limit for mice is missing, but much needed. The existing literature on controlled ammonia exposure seems to suggest an average exposure of about 50 ppm, with peaks in the range of 75 and up to 100 ppm. Finally, our data suggests that housing of 4 mice of this strain at age 12 to 20 weeks for 10 weeks using bi-weekly cage change and aspen bedding does not cause any overt discomfort to the animals that could be causally linked to ammonia building up. At the end, I should mention that, and it's been quite clear, there are significant differences in some of the variables between centers. However, this summary, the main conclusions drawn from the data, applies to all the experimental sites. Thank you for listening.
Okay, that concludes our 3 presentations from our expert panel today. Going to move into our Q&A session shortly, but first a reminder that the webinar will be available on demand for replay or for sharing with colleagues of yours who perhaps were unable to tune in today. So please share that link. You will be receiving it in the next few days, and that the webinar will be on demand for many months at the very least. Now it is time to segue into our live Q&A session with our expert panel, and this is the last call for your questions, so please use enter them into the box that you see on your screen. And if you would just give us a couple of moments, we will transition into the live Q&A session with our guest speakers. Thank you. Okay, everybody, thanks for staying with us. We are now in our live Q&A session. I'm joined by 2 of the 3 speakers that you've just heard from. Unfortunately, our first speaker, Dr. Prins, couldn't join us for this, but I'm thrilled that Brun and Hervé are with us, and they are ready and eager to take your questions. So please keep those questions coming in into the Ask a Question box on your screen, and we will get to as many as we can in the next 15 minutes or so. Brun, I would like to start with you with this first question. You talked about and you presented an innovative new method for ammonia sampling from the side of the cage and mapping the cage floor, which is in contrast to earlier methods. Can you say a little bit more about the difference between these approaches, and does it perhaps mean that the earlier conclusions from some of these earlier papers might be affected by the ammonia sampling methodology?
Well, of course, the methods used will impact the results. So at least some of the variances that we find between studies can be explained by the experimental design. I think it's important here that with the experimental design that we used, enable us to map the ammonia concentration across the cage floor of an IVC through the cage change cycle in some detail. And we could confirm that there is a gradient in the IVC. This has previously been noticed by 2-point measurements, but we could have detailed enough maps to also see a correlation between high levels of ammonia and the location of the latrine. So I think, yes, we provide a more detailed map of the buildup of ammonia compared to previous studies.
Hervé, would you like to add anything to that?
Oh, you said pretty much everything. Maybe some of the studies with only 1-point measurement, the levels that they have is, of course, the average in the cage. So it could explain the difference in the maximum, for example, that we can measure in our cage, because it's not an average anymore. If we measure ammonia just over the latrine area, of course, it's very high. And if you take a method which measure only 1 point in the middle of the cage, which will maybe not be above the latrine, it will be, of course, a lower measurement compared to what we've done. This could explain maybe some of the differences we have with other papers in literature.
And one thing we must remember is that even though we did rather detailed mapping, this is not to be confused with controlled exposure. So there's a distinct difference between experiments where they have used controlled exposure levels of ammonia and those that we infer from a rather detailed map, but nevertheless we infer the levels of exposure. So it's a distinction here.
Okay, thank you. Some good questions coming in from the audience. So this one is very straightforward and simple, perhaps. How many mice can be tracked at once?
Be tracked with some detail with respect to how they position themselves relative to others with the techniques we have used, only one. So the rest of the data comes as a group measurement, not as a measurement of a number of individuals, which we can make distinctions between.
Okay. What was the air exchange rate in the cages? Could we clarify that, please?
Yes, easy to clarify. 75 changes per hour, if I recall correctly.
Okay. Okay, good. Was there a difference in fighting, in the incidence of fighting in males when housed 4 to the cage versus 2 to the cage?
We did not find any overt signs of fighting. And when the study was conducted, we had no means to use the DVC output to track possibility of fighting. I know there's some work ongoing now to derive algorithms so that the output of the DVC can be used to indicate that fighting is ongoing, but we did not have access to that. But there was no overt signs of fighting between them.
Okay. Hervé, jump in if there's anything else you would like to add. But you...
No, not
okay, good. Let's keep.
I speak too much.
No. It's fine. It's totally fine. I'll tell you what. We'll let Hervé answer this one first. Does spot cleaning introduce a more significant variable than a fixed 10-day cage change?
Well, I think it depends on what you're doing. If you're performing experiments with groups in different cages and they are not changed at the same time, I think, yes, it will be a very significant variable in the experiment. I think in this case, you should change all the cages of your experimental group and control group at the same time. We've seen in our paper that it has a huge effect on the activity of the animals. So if you start doing spot cleaning, you can mess up with the result of the experiment. So yeah, it's something to consider in the experimental design.
Okay. Rune, anything to add?
No, I think that's an important aspect.
Hervé, have you extended the cage change interval to longer than the 14-day cycle?
No, we haven't done that in this experiment.
Okay. Do you think cage change frequency can be longer than the typical weekly or bi-weekly frequency?
You want to answer, Bruno?
I think that that's a difficult question to ask. I think because what we discuss in the paper is what kind of improvement can we do to reassure in-cage welfare during prolonged periods of time. And what we arrive at discussion-wise is that to change the area change rates is probably not a very good strategy. We should work along the line to improve the materials that we supply the animals with. Of course, this material must be considered to be something that the animals prefer and like, but it should also then be as atoxic, do not provide a lot of dust and other things, and absorb chemicals like urea in a good way, keep it into the material, and maybe that can improve the period that we can keep the animals without an upsetting cage change.
Okay.
I don't know, Arvid, would you like to add something to that?
No, actually, the DVC can tell you when to do the change in a cage, to do spot cleaning automatically. Of course, if you have only 2 animals in a cage, it will maybe advise to change the cage in 3 weeks or 4 weeks or more. It depends on the bedding and things like that. But yeah, like Brun said, it really depends on what you put in the cage for these mice. Theoretically, yes, you could, have a longer change cycle, of course, but there are many aspects to consider there.
Well, speaking of what you put in the cage, 2 questioners want to know about the impact of the different strains that you have or could look at. Have you examined other strains or stocks of mice, and are the results applicable to other strains?
I have no data on that in my core facility. I don't know if Brun, you've performed some experiments.
But it's probably a very important factor. I mean, it's not only the sex, the number of animals. We refer also to the importance of the biomass, and strain is certainly an important aspect here to consider. And I would be surprised if different strains would show the same dynamics as this particular strain that we used here.
Yeah,
being a very common strain, but nevertheless, the results are constrained to this very strain.
very good. What is the consideration behind your assessment that higher activity level after a cage change should be defined as hyperactivity, as if you conclude the higher activity levels should be analyzed as negative? I don't quite understand the last bit, but could we talk about that?
That's a good question, yeah. With the DVC, we have more questions than answers, I think. But it's good because we learn so much about activity of the mice in the cage. It's a good question. It's what is good for a mouse? Should it be reactive or should it be resting in a corner of the cage? It's a good question. But still, if you look at the results we have, you can see that the rest activity during the daylight is perturbed, which is not normal if we consider the same thing on our side as human. We don't like to be awake during the night because the resting period will not be good enough if they are active. So that's why we consider that high activity after a cage change is not really good for the animals. But again, maybe more studies are needed to really confirm that. And what is an happy mouse is difficult to say according to the locomotion activity.
Yes, one comment on that is that we published more recently another paper looking at rhythmicity of activity in rest and sleep of mice of this strain. in IVCs. And what we could see then is that the normal active sleep pattern during the daytime is disrupted for several days. So they don't have the same amount of long rest and sleep as they do 4 or 5 days after a cage change. So it disrupts the sleep and rest pattern for sure. And if that's good, I doubt, but the overall activity level is much more difficult to use.
Okay. A few more questions. Home cage activity, as we've heard, gradually decreased over the 2 weeks following a bedding change. And this question asks, is this an artifact of the sensors and bedding moisture content, or are the animals truly changing their behaviors even 5 or more days after a bedding change? And if they are changing their behavior, what are they responding to, do you think?
So I think I can start to answer this, and Harvey, you can fill in. Now, and this was noticed early on. So what we use as activity is an electrode activation, and that's during a change in capacitance between records. So we have 4 samples per second. And that means that the sampling rate of activations is much, much higher than any bedding of the bedding material can compromise. So that issue is resolved when it comes to the activity pattern that we get out of the DVC. Herbie, I don't know if you would like to comment further.
No, I think you covered it pretty much. Yes. I guess, yeah, you have to be careful about the bedding, of course, and especially maybe not only the moisture of the bedding, but the thickness of the bedding above the antenna, because the mice they used to move the bedding around, you know, and this could be a bias, but that's why we have many cages in our group to average that. But we have to be careful about that. Yes, the thickness.
Yes, the same thing. If we put a lot of stuff inside the cage, like small red houses and so on, and they do a lot of activity on top of these, they will not be faithfully recorded by the electro sensors. Mm-hmm. So that is something you need to consider as well in the experimental design.
All right, we have time for a few more questions. If in the audience you have a question for either of our 2 guests, please enter it now before we close today's webinar. Did your study compare the activities of weekly versus bi-weekly cage changes?
Yes, in a smaller subset of cages on one of the sites, we compared the density of having 4 males to a cage with the density of having 2 males in the cage. We also combined that with 2 different setups using corn cob and aspen chips as the bedding material. So the already mentioned by Hervé, the ammonia levels when you have some 2-2 cage is negligible across 2 weeks, and there's no major difference between the choice of bedding materials either. So I think that what you may want to know is if you can grade the output from the DVC to read the number of animals and their activity. And the problem here is that the output from the DVC is a group output, and there is some relationship between the level of group output seen across the circadian rhythm and the number of animals inside the cage. But it's not a very linear and straightforward interpretation of the data. Hopefully this is an answer to the question. I don't know.
Okay. It seems, next question, it seems that in some cases the living space in the cage floor decreases across the period. Do you think the cages are too busy in terms of the number of mice?
Well, we have to define what's a living space in a cage, because if you're talking about the space taken by the latrine area, it's a living space. So of course they might avoid it because they don't really like to spend their time there, but it's still a living space. So I'm not sure that the cage floor is decreasing across the study, actually. I don't know if you want to add something, Brun, but it's part of the living space. Yeah.
Okay. Would the presence of different environmental enrichments change cage activities, such as resting times?
I would say yes. I've performed some experiments that are not published yet, but yes, according to the enrichment, according to the nesting material you put, or the other kind of enrichment to hide or things like that, activities could be very different during nighttime or during daytime. And it also depends on the mouse strain, but there is an incidence on that. Yes, absolutely.
Okay.
I know that in the facility, for example, there are some nesting material we don't use anymore because the animals they don't use it. Their activity is very different from others, and we consider that it was not that good for the mice.
Let's just quickly go back to ammonia. Can we just briefly talk about ammonia limits? It's 25 ppm is usually the reference. Is this still correct? Is there a real limit for that, and how do we collect it?
Well, the 25 ppm that you usually hear about comes from the exposure restriction for humans at the workplace during 8 hours workday.
Yeah.
So the average should correspond to 25 ppm, and the peaks are not exceeding 50 ppm, but they are not based on small rodents at all. And they are mainly based on self-reported discomfort, cough, coughing, ear tears, and so on. And that's tough to record from mice. So in controlled exposure studies, it is unclear that these limits are relevant, in fact. And I think that if we try to do some kind of systematic analysis of those studies that have used controlled exposures, we would end up with a probably more likely level of exposure limit on average at 50 ppm rather than 25 ppm. And peaks at 75 to 100 ppm, according to the limited number of studies done with controlled exposure. So that is something that we should really address and try to do systematic multi-center study on. We're clamping the level of ammonia, so we actually know that they are exposed to 25, 35, 50 ppm, not inferred by indirect measurement. So that is missing, and the 25 ppm comes from humans.
Okay. Urvi, anything to add on the big question of ammonia detection?
No, I totally agree with that.
Okay, and finally, I think we have come to our last question, back to the impact of cage changes. This seems to be a dramatic stress event for the animals that can last many days. How do you suggest handling this in a standard vivarium? Should one try to limit it as much as possible for non-experimental cages, but then keep it fixed for experimental ones?
I think that could be one approach. Yeah, like I said previously, it actually should be considered in the experimental design, carefully, and yeah, the less we perturb the mice, the better, of course. And as you've seen, sometimes it takes more than a week to get a plateau in the activity of the mice. So if you change these mice again the next week, so you will keep up with a high level of stress. And we haven't measured anything like corticoids or things like that to check what was the stress level at the biological levels. But still, I think twice, one every 2 weeks, it's really a good time, a good cycle to let them rest at least for a week before you perturb them again. I would say. I don't know if Brun, you want to to add something to
I agree. I think it needs to be controlled for, so it must be built into the experimental design. So if you decide to do the testing, let's say 3 days after a K change, you must control for it and do it on another day and see if you get a different impact depending on the day in relation to the K change day. So I think that's something we need to build into the experimental design in the future. But that depends also, of course, on what kind of study you are conducting.
Okay. Well, that's a very good place, I think, to leave our discussion. Fantastic response from our audience. Well over a dozen great questions. So thank you very much. Today's webinar will be available on demand shortly, either for repeat viewing or for sharing this valuable series of talks, presentations, and discussion with your colleagues who perhaps were unable to attend the live broadcast. My thanks to all of our distinguished speakers, including those not present here, and to our friends at Technoplast for yet another successful and very interesting webinar. Thanks to the Marianne Liebert Multimedia Group, led by Liana Jabs, and our friends at Workcast for helping produce another seamless webinar. And most of all, thanks to all of you for watching and being so supportive for our webinar program. I'm Kevin Davis. Thank you for watching again, and goodbye for now. Arrivederci.